Okay. So much fun to be here and, be talking with an audience quite different than I normally do. So I'm looking forward to your questions. I expect them to be quite imaginative and different than I normally get. So please don't hesitate to ask me questions as we go along. So I'm going to talk about epigenetics of human health and disease. I'll start with an overview of what epigenetics is, some of the basic mechanisms that were first discovered, and then we'll go into a few vignettes from my own work. So what is epigenetics? Very simply, epi translates, of course, as on top, from Greek, on top of genetics. So epigenetics are phenotypic changes, and that generally means gene expression changes and all the sequelae after that, without any genotype differences, any DNA mutations as we, whoops.
What's a sequela?
Oh, what does sequela mean? All the downstream effects. So phenotype starts with gene expression changes in the genome, and then those genes that are altered up and down then lead to what we observe as changes in development, behavior, what have you. Okay, everybody following that? Sorry about that word. All right, so another way of, so that's a word, epigenetics, but now let's take a look at the clear manifestation of epigenetics in an organism. And here we'll use a mammalian organism as an example. So development in humans, starts with one cell, the zygote, the union of the sperm and the egg, leading to rapid cell division, and then a fetus, which even at this point includes about 200 different major cell types and a lot of other additional cell types. So that is a clear manifestation of epigenetics. We start from one single cell, which has one genome, and that genome then, through this developmental course, then has to assume at least 200 different phenotypes. All right, so I think that's a pretty clear manifestation. Now, this idea of epigenetics, indeed, the word epigenetics was coined by Conrad Waddington some 50, 60 years ago, a visionary developmental biologist who drew this, this diagram, which you might have seen at other times. So he drew this landscape, this epigenetic landscape, putting at the top of this landscape one single cell, so a totipotent cell, a cell capable of differentiating along this epigenetic landscape into these 200 different phenotypes. So that really interesting landscape we can now translate, through the next 60 years into this idea that at the top of this landscape is a stem cell or an embryonic stem cell, different types of stem cells, and through epigenetic alterations then take on these different cell types, all the cell types that we find in our soma. All right, so then we can think about disruption. I want to tell you about not only normal epigenetic regulation, but also disease. We can then think about epigenetic disruption as being at the heart, or at least partially at the heart, of disease such as cancer. All right, so now we would like to know what are these epigenetic changes. So now we can look at the DNA and the packaging of DNA into the nucleus. The DNA strand is about 3 meters in every one of our cells. So obviously the nucleus is a tiny, a couple of microns, or whatever, 100 microns. So this DNA, this 3 meters of DNA, has to be packaged in every cell, the nucleus of every cell. And that's accomplished through the packaging material that are called proteins called histones. And they two each of these form the octamer. Here you can see it here with the DNA wrapped around it. And this complex of DNA plus histones is called the nucleosome. So that's the basic lowest order of packaging in the eukaryotic genome. And so now we can look at the structures, including the simple structure of a single nucleosome, and then all of the nucleosomes assembled on the DNA as beads on a string in the electron micrograph shown here. And that's about 11 nanometers in width. Now the next order of packaging, well, it used to be called the 30 nanometer fiber, shown here, but this fiber probably does not exist in the cell. But there are some sorts of packaging that occurs, some sort of higher order packaging. Now one method of packaging are enhancer promoter loops. We'll talk about those later. They form these larger structures, and then these loops and other sorts of interactions form what are called TADs, topologically associated domains of DNA, and then other sorts of interactions form phase-separated nuclear bodies, and these are very large complexes then. And finally, at the highest order of packaging is the metaphase chromosome, that occurs only during cell division. All right, so now if we go back to this slide of this nucleosome, we can now take a look at the different sorts of chromatin mechanisms that exist to package the genome and to regulate the genome. And so at the lowest order of modifications are DNA and RNA modifications. And then there are post-translational modifications of the histone proteins. There are non-coding RNAs within the genome that carry out various different regulation. There's nucleosome remodeling. These nucleosomes can be altered in their location and indeed their composition through enzymes that can exchange histones and change the actual location of these nucleosomes. And then I referred to a moment ago, three-dimensional architecture, these large-scale loops, and these TADs that can form in the DNA. And then finally, an area of great interest that's sort of blossoming right now in chromatin regulation and epigenetic regulation are nuclear bodies. Some of these have been known for a long time. The nucleolus I'm sorry, I didn't define these. These are membraneless bodies within the nucleus that form and are stable. Well, in the case of the nucleolus, it's stable, where ribosomal RNA is made but currently there are a number of other nuclear bodies that are being discovered. I'm not going to go into those. They're really interesting, and indeed, all of these areas, I'm going to really focus on these. There's not time really for me to focus on all of these levels of regulation, but I'm happy to talk about these in questions or over, later on. I'll stay for this evening, so I'm happy to discuss any of these areas that you're interested in. All right, so let's start with DNA modifications. They're really the first level of epigenetic chromatin regulation that was discovered. This shows how DNA methylation occurs. Most of methylation occurs on cytosine side chains in the DNA. Here's the base, the cytosine base side chain. And DNA methyltransferases use S-adenosyl methionine as a donor group to methylate this carbon at this 5 position in this cytosine ring. So here's it's a very simple modification, methylation of cytosine in DNA. All right, so let's pause for a minute. I want to, yes? Absurd?
We learned that methylated cytosine is prone to mutations.
Mm-hmm.
And has there ever been any evidence that it's sort of intentionally methylated so that it might be mutated somatically? It could not be an epigenetic relation, you know?
Thinking about that, I mean, it's certainly methylated. I mean, there are mutations that involve methylation, intentionally methylated for mutations. So I guess maybe you're talking about something like recombination of, during in immune cells, something like that. Is that what you are referring to? That could happen.
That would be an explanation, but I think one of the things that's a consequence of the high mutation rate, which is about tenfold more than cytosine, as a spontaneous mutation rate, is the dinucleotide methyl CpG, right? CG dinucleotides are actually very rare in human DNA. They're rare because they mutate in that way, but that is one thing to think about. There means the signals change with time, or in people,
No.
in various lines. So it could be generating diversity at a higher rate than other things.
No.
And it's certainly the most common mutation in cancer.
No,
So,
that is true.
yeah, but I don't think it's there on purpose.
Yeah, I think purposeful. The only case you would see that is if it were going to be used for, say, VDJ recombination, something like that. So it's located there. It causes a mutation in the sense it's causing a rearrangement of the DNA. Something like that you could think about. All right, so I'm going to pause for a second and give you a very simple, yes?
Sorry, could you go back one slide? Yeah, my question is about SAM. So I guess SAM is a downstream molecule of the methionine, right? So my question is that, so for DNA methylation to occur, does the cell needs a lot of methionine to be present? Or, I mean, if methionine is a limiting nutrient for the cell.
No, it's not methionine before it's methylated. It's acetyldenyl methionine is the donor of the methyl group into the DNA.
Yeah, but biosynthesis of cell needs methionine, right?
Mm-hmm.
So if cells, so if cells is limited by methionine, maybe cells don't have a lot of SAM to power this reaction. So I'm just wondering, so for cells to have DNA methylation to occur, the cell has to have a lot of methionine so that it can synthesize SAM, so that it can power the reaction. So there's a correlation between DNA methylation and methionine concentration?
You mean cells that have high levels of methionine?
Yeah,
have high levels of
methylation.
Well, I think there is a metabolic. Well, I think we're going to get to that next next lecture. I think that high levels of methionine need to be present in cells that are going to.
Okay. Yeah, I was just wondering because I saw SAM is a cofactor of this reaction. Just wondering if Okay.
Yeah. All right. So, whoops. Looks like I'm not going to get to my lecture. Yeah.
Chemistry question. So all these reactions are happening in equilibrium. So it's going back and forth, methylation to methylation, or is something that happens once?
Okay, I'm going to get to demethylation in a little while.
Right, but it's happening in equilibrium, so it's going back and forth. It's just one.
It's not a simple demethylation reaction. There is no simple demethylation, so it has to go through a very elaborate. I'm going to get to that in a second. If you just wait, I'll get to that. Okay? All right, so let me just tell you a little bit about a simple example that illustrates the importance of epigenetics in human health. So if we think about identical twins, they have an identical genotype. They share a precisely identical genome. So at birth, the same genes are turned on. The same genes are turned off in each of the identical twins, and that uses DNA methylation. Perhaps we'll get to it in a second, what it does, and other modifications to turn genes on. Okay, now through diet, the environment, aging, Arnie referred to some of these things a moment ago, Identical twins can vary. So here's an example of two identical twins. They were both, Dear Abby and Ann Landers were advice columnists. They both had exactly the same job. They must have been pretty similar intellectually to do that weird job. So they died 11 years apart, and indeed one died of cancer, the other died with, with Alzheimer's disease. So in one twin, the cancer genes, in a simple example, were turned on, Alzheimer's disease genes off, and in the other twin, the opposite. So over their lifetime, even dying 11 years apart was just pretty substantial, considering the human lifespan. Okay, so this means that while genetics, I'm not disputing, genetics is crucial. Much of our biology, our individual biology, is determined by genetics. Epigenetics is also important to your health. And epigenetics is malleable. So that's the nice thing about epigenetics. Genes are hard to fix. You're hearing, I'm sure, about CRISPR technologies, and we're certainly thinking about those methods now, at least somatically. On the other hand, epigenetics involves enzymes, as you'll see in a little bit, and they can be manipulated and regulated. All right, so now let's look at methylation again. Methylation occurs at CpG dinucleotides, as Arnie just mentioned. Here you see them. So de novo methylation, let's say you'll see in a moment at the beginning of life, it occurs through certain DNA methyltransferases, a certain subset of them, whereas other DNA methylases have the ability to maintain methylation. So CG dinucleotides are, of course, palindromic, and so these methylases have the ability to recognize methylation in this hemimethylated form and then place methylation on the other strand. So that restores methylation. So you can now imagine through cell division, as the DNA is synthesized, you can restore methylation in the cells that result. All right, so epigenetics then has this additional idea of heritability, that it is very important in maintaining cell identity, for example. So maintenance methylation provides a mechanism for inheritance of DNA methylation during mitosis, which would be as cells are specified to maintain those cell lineages over a lifetime, and during meiosis, when you create new germ cells. Now, how does that occur? We're going to talk in a minute about reader domains, or domains within these enzymes that recognize their own modification and are then able to put it on the other strand. That got messed up a little bit. All right, so now let's talk a little bit about the function of DNA methylation. Have you already talked about this? Yeah. Okay. All right, so what is DNA methylation doing? It has a number of different activities. For one, it controls gene expression. It's found over gene bodies. So these little black lollipops are DNA methylation. And you can see over this gene body, these exons, you can see there's a lot of DNA methylation. So in order to maintain homeostatic levels of gene expression, DNA methylation is found over the transcribed regions of DNA. It's found over transposable elements and repeats. The genome is composed of, of a huge amount of DNA, of transposable elements, active and inactive, and repeats. And it's very important to control their expression and their transposing. So these elements tend to be highly methylated. And where is it low? It's low at promoters. Here you can see CpG islands, the open lollipops. That represents low methylation, and yet very high concentrations of methylation of CpG residues at promoters. Indeed, there's a high concentration because DNA motifs for transcription factors tend to be high in C and G, and so it's very important to maintain low levels. And it is thought, although not proven yet, that there are activities to maintain low methylation. It's not really understood. Even though DNA methylation is the longest known modification that we're going to look at today, it's amazing how there's still mysteries that abound, and this being one of them. All right, so that's the function. Now, this is a very interesting modification with respect to genome inheritance. So let's take a look. This is during embryonic development, and this is the level of CG methylation. Okay, so let's start with germ cells over here on the left, sperm and eggs. Here's the zygote. So the level of methylation is very high in germ cells, and that's to protect the genome. So DNA methylation tends to disallow transcription factors from binding, and so you find that it's very high in germ cells, and as I said, across gene bodies. So that allows a protection of the genome, also transposable elements, very important in germ cells and through early development to prevent transposable elements and repeats from being expressed. All right, so then in very early development, here's the blastocyst. There's erasure. So that erasure is very important to reset the genome during early development for the next generation. It is utilized even in early development for parental imprinting of key growth genes. So you get a specific methylation pattern of certain genes from your mother and from your father, and that regulates some very important growth genes. And indeed, if that's disrupted, you see growth, you see human health disruption in overgrowth and undergrowth. Okay, so then in somatic development, now you see DNA methylation being restored. That's very important for cell type identity. Okay, and then in primordial germ cells, you get a second wave of erasure. That has to be very low again to set the correct methylation level in the germ cells. Okay, so with respect to DNA methylation, there's no simple here are the DNA methyltransferases leading to 5-methylcytosine. There's no simple method. There's an enzyme class called TET enzymes, TET 1, 2, and 3. I don't want to point to these. They all do the same thing. They oxidize this methylation into oxidized forms, hydroxymethylation, and some further forms. This is still not demethylated, this oxidized form. In fact, it requires decarboxylases and base excision repair to reset unmethylated cytosine. So there's no simple equilibrium in this case. All right, now cancer mutations affecting DNA methylation, there are a lot of them. This is already from 2012, I believe, this review. Lots and lots of mutations have been seen in DNA methyltransferases and these TET enzymes. And I want to mention also that this, what you find, I mentioned before, in order for DNA methyltransferases to restore methylation, it requires this PWWP domain. I'm going to get to this in a second, the general class of reader domains. So this domain is present in this DNA methyltransferase to allow, this is the maintenance type then, to allow restoration of fully methylated DNA after cell division. All right, so what sorts of mutations do we see? We see hypermethylation of tumor suppressor genes and microRNAs. Indeed, I made a note to myself, this is what was first found in cancer, hypermethylation in the promoters around tumor suppressor genes, and that led to a general model that methylation occurs in promoters to regulate genes, to turn them off. And that turns out to be exactly the opposite of what is actually the case, where I told you a few moments ago that CG islands are unmethylated. In fact, there are activities that maintain them in an unmethylated state. But in cancer, you see hypermethylation of the promoters around DNA tumor suppressor genes, and that leads to their lowered expression, and that's a bad thing. You also see hypomethylation of oncogenes, high levels of oncogenes then, broad dysregulation across the genome over gene bodies, over transposable elements. And then you also see some very interesting mutations in certain enzymes, such as IDH, that regulates, that normally makes it's in the metabolic pathway to make cofactors for DNA and histone methylation. And so these neomorphic mutations in IDH lead to what's called an oncometabolite, a metabolite that cannot donate, it's sort of an end suicide pathway in metabolism that then lowers DNA methylation. Yeah.
When did tumors start high at those times?
Yeah, it's a great question. I mean, part of it is this is old, and so these were the first to be found in blood cancers, which were the first to be sequenced. Now we know that they're present in other cancers as well. All right, so I'm going to very quickly mention RNA modifications. Very quick little vignette here. RNA modification, there are 160 types of RNA methylation. It's a brave new world. I'd like to give you a little bit of forward-looking ideas as well. It's been known for a long time, but with much better mass spec technologies, we now know there are 160 different types. And we'll get to histone modifications. That's also bewilderingly complex as well. Now, RNA modifications are found in mRNA. They appear to regulate initiation, splicing, stability, and translation. They're very prevalent in tRNA, where they regulate translation via stability and interactions with mRNA, with the whole ribosome. Ribosomal RNAs, regulating ribosome structure and translation efficiency. And in cancer, they're already being identified as the enzymes are mutated and overexpressed. So this is a very interesting area that's going forward. All right, so now we'll move on to histone modifications. And I'll spend a moment describing how these were discovered. It's a very important area. All right, so this is the famous structure from Tim Richmond's lab. Carolyn Luger and Tim Richmond solved this structure in 1997. And it shows the X-ray structure of a half of a nucleosome, cut in half. So here's the longitudinally. So, the cross-sectionally. So here's one wrap of DNA. There are actually two wraps of DNA around the nucleosome. You're looking at one. There's 146 base pairs of duplex DNA that wraps around, as I mentioned earlier, a tetramer of two. The nucleosome is built up in situ with two dimers of H3 and H4 that's laid down on the DNA, and then two dimers of H2A and H2B come on to complete the nucleosome. And this is the structure. It's really quite beautiful structure, and it's really informed a lot of work in the field of epigenetics and chromatin regulation in these now, wow. Is that 20 years? That's scary. Scary stuff. All right, so now let's take a look at the histone proteins. These are the four core histones, H2A, H2B, H3, and H4. Here you can see that they're composed of a globular, highly structured domain. That's what's inside of the nucleosome. And then there are extended amino and carboxyl terminal tails. So actually, if you just look back here at the structure, the globular domains were beautifully, they're a nice repeating structure, so they were beautifully visualized in the X-ray crystal structure. However, the tails, as I said, are unstructured. So they're not easily seen. You just get a little bit of a couple of the tails in this structure that are enough, they have enough repeat that you see them in the crystallization. All right, so here are these tails. So now I'm collapsing the globular domains in the middle here and extending the amino and carboxyl terminal tails and showing you the bewildering variety of histone modifications. So whereas DNA modifications are very simple, there's really just cytosine methylation, and now a little bit of adenosine methylation has been seen in certain organisms. But for histone modifications, you can see there's lots and lots of them. So in green is acetylation, in blue is methylation, in orange is phosphorylation, ubiquitination, sumolation, and there's something called isomerization. So you can see there's lots and lots of these, and you might say, how can this, how can any sense be made out of this? So now if we take a look at a very old picture of the cell, of the eukaryotic cell, you can see here's the nucleolus in the middle, and you can see that in this staining, DAPI staining or dark staining pattern, you can see that around the edge of the nucleus and around the nucleolus is very dark staining so-called heterochromatin, and in the middle is light staining euchromatin. All right, so that's interesting. Now flash forward, what we know about this is that euchromatin is open regions of chromatin that are transcriptionally active. Heterochromatin are closed regions that are transcriptionally inactive. So now if we use this as a backdrop, we can now make sense of this bewildering variety of modifications, looking at how they correspond to this opening and closing of the chromatin. So on the left here is open chromatin represented by these cylinders of nucleosomes. Acetylation is opening to the chromatin. It tends to open it up structurally by electrostatic changes to the histone, to these tails. And methylation, in the case of this particular, I'll show you these a little bit more as we go along, these specific types of methylation. This type, on histone H3, lysine 4 trimethylation, I don't want to get into the details here, a certain type of methylation is found at active parts of the genome at promoters that are where genes are active. So that type of methylation is activating. On the right here is compact chromatin. You can see the nucleosomes close together. Here you see low levels of acetylation. Acetylation tends to be activating, but you see high levels of other types of methylation on histone H3, lysine 9 methylation and lysine 27 methylation. Well, I'm summarizing in a second work that took place over about 20 years of figuring out the correlation of the enzymes that carry these out, what they're doing in the genome, how they're localized, how they're brought in to specific locations. A lot of our work from my lab, early work from my lab, studied the acetylation, the structures, the enzymes, what kind of complexes they're in, how their X-ray crystal structures, et cetera, et cetera. So tons of work, and I'm not going to go into a lot of that work. All right, so histone and factor modifications in cancer, lots of them. On the left here, again, this goes back quite a long way. So 2012, I took these from a review, so you're not going to see all of the different cancers and types of modifications. There are lots of mutations that are seen. So here are a whole group of mutations in histone acetyltransferases. They're actually called CATs for lysine acetyltransferase. We're going to look at a couple of these in a little bit more detail. some of the reader, I'm going to get to this on the next slide, I believe, reader proteins that bind to these modifications. And then on the right, methyltransferases, lots of them, and they're reader proteins. Now, I just wanted to mention a very interesting, another sort of oncomutation that's been seen in glioblastoma, pediatric glioblastoma. This mutation that's seen in the histone itself, histone H3 lysine 27, is mutated to methionine. That is a really interesting sort of oncomutation. It's rare. It's only found in pediatric glioblastoma for reasons that are not understood at all. And even its function is a little unclear, but it's thought to be a suicide modification, trapping the enzymes that methylate this, this residue, trapping them into either sequestering them so that they're unable to methylate where they need to be carrying it out, or changing their structure to an inactive state. Not clear exactly what's going on there, but very interesting, big, lot of work being done in that area. So just wanted to mention that. All right, so this is the summary then for all of this work I've been telling you about. We have lots of enzymes, and they're called writers, you know, colloquially, of kind of a convenient way of thinking of them. The enzymes, the DNA methyltransferases, the histone acetyltransferases, the lysine methyltransferases, they place these modifications, and again, they're placing them via recruitment by, say, transcription factors and other ways of getting to their important locations. So they're kind of dumb, you know, they don't know where to go in general. And so the location is provided by transcription factors in other locations. The enzymes that remove these modifications, we talked about TET enzymes. They're also histone deacetylases and lysine demethylases, like the TET enzymes I was telling you about. I'm sorry, there's yet other groups of enzymes. We can call those erasers. They remove these modifications. And then we can call the effector proteins that bind to these modifications and carry out all of the mechanisms downstream, the sequelae, I used that word earlier. So these things are just modifications. They may be changing the structure, but another thing that they do is they bring in all of these reader proteins that then carry out all of the mechanisms downstream of these modifications. And so there are hundreds of these enzymes and hundreds of these effector proteins that can bind to them, providing unparalleled opportunities for therapeutics. So therein lies the kind of the gold for human health. Yeah.
So just to be clear, in terms of like choosing where to methylate,
yeah.
Saying it's more written in the actual like DNA codes and factors there, or is it?
Yeah.
The things? Okay.
Yeah. So for a lot of what's happening here is happening around genes. And so transcription factors will bind to enhancers. They will then recruit through protein-protein interactions these enzymes and their complexes to lay down the right modifications for regulating those genes, either on or off. Over gene bodies themselves, it's a much more complicated, where RNA polymerase goes through many different modifications as it moves along the transcribed region. Its modifications then lead to various different modifications over the genome. I'm going to show you a little bit of that as we go along. Okay, so I don't have time, as I said, to talk about it. Very interesting areas of ATP dependent. There are enzymes that use ATP and can change the location, remove these nucleosomes, these histone, octamers, and can change the composition, using histone variants that are specialized for setting up the transcription, setting up the genes, setting up the promoters. You know, again, a lot of work and a lot of interest in this area as well, and mutations in cancer and these enzymes that lead to this. Non-coding RNAs are present, whole area unto itself. Some of them are altered in cancer. And then you get to three-dimensional architecture of the chromosomes, the so-called TADs, for those of you that follow this area. Very interesting. Still, you know, brave new world in understanding what these big genome structures are all about, how they're regulated, what their mechanisms are, if they actually exist. We can talk about that later. I have my own opinions. And then nuclear bodies that I mentioned. Nucleolus is a famous one. Lots of other nuclear bodies that are fascinating to study. And then new areas just emerging of enhancers forming nuclear bodies, heterochromatin forming nuclear bodies. Those are brand new in the last few years. Very interesting areas. We can talk about that over dinner or whatever. All right, so human health. Epigenetic regulation involves enzymes, DNA and histone modifying enzymes, chromatin remodeling enzymes. These are frequently mutated or overexpressed in disease, and they are superb targets for drug development. There are DNA methyltransferase inhibitors in the clinic, HDAC inhibitors in the clinic. This is an effector domain, called a so-called bromo domain that binds to acetylated histones. There are inhibitors to these domains that are in final phase 3 clinical trials. All right, so now I'm going to go on to 3 vignettes. We'll see if I get through them all. Epigenetic pathways in human health and disease. These are 3 areas from my lab. My lab, the kind of maybe great thing and maybe seductively destructive thing you might say about epigenetics and chromatin regulation is you can study it in many different areas. So in my lab, we study cancer, we study the brain, we study aging and neurodegeneration, and I'll mention in a little bit, we also study ant behavior. And lifespan. I'll show you in a second why, or a little bit later. All right, so we'll start with cancer. I'm going to tell you a little story about mutant P53. It acts as an oncogene. When it acts as an oncogene, it utilizes epigenetic pathways to drive disease. All right, so wild-type P53 is tumor suppressive. It's protective to stress and DNA damage. Via surveillance mechanisms in the genome. Its function, by and large, is a DNA binding transcriptional activator. It binds to genes, turns them on, which turns on key downstream pathways involved in cell cycle arrest to then allow DNA damage repair, and if that doesn't work out, suicide, apoptosis of the cells. All right, so how does P53 work? Well, we'll go through this step by step. In response to genomic stress, damage to the DNA of various types, or activated oncogenes, a signal transduction cascade ensues where P53 is phosphorylated by kinases or acetylated by some of these histone acetyltransferases moonlight as transcription factor acetyltransferases. P53 then becomes nuclear localized, it becomes activated, it forms a tetramer and it binds to DNA to turn on genes involved in those pathways I mentioned a moment ago. All right, so P53's normal role is to restrict cell growth. Its normal role is a tumor suppressor to prevent cell proliferation. However, it's mutated and overexpressed in more than 50% of human cancers and behaves as an oncogene. In fact, Arnie discovered P53. David Lane is also credited with discovering P53 and Arnie can speak for himself, but they thought that P53 was an oncogene. They saw that it was overexpressed in cancer, and they didn't realize that, that it was actually a tumor suppressor in its normal role. Good summary, Arnie?
Yes.
All right, good. So,
9 years of my life.
yes, I know. I know. And beautiful, absolutely spectacular work. All right, so what are the abnormal mechanisms when P53 acts as an oncogene? And this has been for the last 20 and more years, it's been a huge area of investigation, and our part is here, that I'll show you now. All right, so P53 is probably, by the way, is the most published protein and gene, about 100,000, I would say, papers on P53. So here's his domain. I don't know, there may be other things approaching it now, I'm not sure, but it's slowing down a little bit. But here's P53's domain structure. It has a very large DNA binding domain in the center, 2 different activation domains on the amino terminus, and a tetramerization domain and some regulatory domains on the carboxyl terminus. Now, so P53, you see hotspot mutations when it behaves as an oncogene, and there are about 6 of these mutations that you see very prevalent in cancer. These missense mutations occur in the DNA binding domain, and they lead, again, as I said, to a very high level of mutant P53. And this DNA, its conformation is messed up in many cases. It's unable to bind directly to DNA. So cells with these hotspot mutations, I'll point out, do not act as just a null, because if you make a mouse that it's lacking P53, the mouse will develop cancer, but it's going to take a while over its lifespan. But now if you put in one of these hotspot mutants of P53, now it develops cancer very, very rapidly. So it's really a gain of function of P53. All right, so we wanted to know whether this gain of function, these hotspot mutants of P53 might bind to the genome. I just told you that it can no longer, it's mutated its DNA binding domain, it can no longer bind on its own. But we wondered whether it might nonetheless, there's a high level of this stuff in the nucleus. So whether it can bind to the genome, and what can this tell us about tumor mechanisms. So we carried out chromatin immunoprecipitation sequencing mapping of gain of function P53. Do I need to explain seq? They've no? They don't know. Ooh. No kidding. Ooh. All right, so if you want to know where a protein binds within the genome, you can map its location genome-wide via the use of an antibody that binds to that protein. So in the case of P53, you have an antibody that binds to P53. You can float that antibody into cells. It gets into the nucleus, and it binds to its target, in this case P53, and now you cross-link all of that material together. You isolate the DNA with the factor still bound, the protein still bound in the antibody, and now you break up the DNA, and you capture the material that's associated with P53, and then you clone that essentially and sequence that. Okay, and that then gives you a genome-wide map. I might have messed up a little bit of that, but that's essentially the bottom line. And now you get a map across the entire genome of where P53 is bound. You can do that with any factor you like in the genome, provided you have an antibody that works well. And that isn't easy. You have to prove that it's good with a lot of controls. All right, so if we do that with wild-type P53, we see that wild-type P53 binds to its key target, the promoters of its key target genes. So I told you earlier that it binds to some genes that lead to cell cycle arrest, P21, or PUMA, an apoptotic gene, And this is what's called a genome browser. And so you're looking at the gene structure here, the exons and the introns, and we can find the promoters with gene annotation, lots of work done by many people like you that have gone into this sort of work over the last decade or two. All right, so wild-type P53 binds to the promoters of P21, MDM2, and PUMA, very important genes involved in cell cycle arrest and repair, et cetera, and these and others. All right, now this is wild-type P53, but the substitution mutations, these hotspot mutants, do not bind to these locations. We do not see them there. However, if we now look at where they bind, we find that they bind to other locations. In fact, we found that they bind to a number of different epigenetic target genes. So here I'm showing you one called MLL1, another one called MLL2. Now, P53 can't bind to these locations, so we found, and I'll give you a little summary at the end, that it's binding indirectly through its association with a transcription factor that recruits it to these locations. So what about these genes? Why are they interesting? Okay, well, these code for the MLL1 and the MLL4 proteins. They methylate here, histone H3, at lysine 4, here at this location. And these are very interesting locations. They are very important in methylating promoters in the case of MLL1 and enhancers in the case of MLL4. And those are important in turning on these genes. And it turns out that that these target genes, their target genes are genes very important in setting up early development, such as I'm not going to go through it, but Hox genes. These set up the patterning of embryos, pattern development, and these genes are upregulated in cancer, and their target sites are, we found that are highly upregulated in these gain-of-function P53 cancers. All right, so then we could ask, is the upregulation of these genes involved in tumorigenic growth in gain-of-function P53 cells? Yeah. Why what? I'm sorry?
Why the mutant P53 is binding to these to a transcription factor and the
Right.
type is not binding there?
Right.
What is happening?
There's another transcription factor that's binding to these genes, and it's recruiting
I see.
P53.
Why is recruiting the mutant and not the wild type?
Yes, it will recruit the wild type, or you can see an interaction, but it's very weak unless P53 has, its conformation has been altered, or its DNA binding has been, its own DNA binding has been suppressed by these mutations. Yeah.
So this is an example of how mutated P53 can upregulate specific genes that are usually upregulated. Does the mutated P53 also downregulate specific genes as well?
We don't think so. We don't think P53, P53 has these very potent activation domains. So it really, its major role is as a transcriptional activator. And indeed, we and others know that this function of P53 requires its activation domain. So it appears to be acting primarily as a transcriptional activator. All right, so if we knock down MLL, we can see that in these gain-of-function P53 cells that are growing very rapidly, if we knock down either of these two genes, we see that the growth of these cells collapses. Other cancers that have wild-type P53 are not altered by the knockdown of MLL1 and MLL2. And indeed, this change in growth is a phenocopy. It is very similar to if we knock down P53 itself. Indeed, we can see that this MLL knockdown leads to reduced cancer growth, reduced tumor growth in a mouse model of cancer. So you can see when we knock down MLL1 compared to a control knockdown, you see much smaller tumors. All right, so here's the mechanism that we believe is going on here. So mutant P53 cannot bind to its target sites. It's lost the ability to do that. Instead, it binds because of the disruption of its interaction, its own ability to bind DNA, or through a gain of ability to bind to other transcription factors. We think ETS is a major transcription factor that's recruiting P53. It's recruiting it to this MLL1 and MLL2 genes, turning them on, leading to MLL1 and MLL4 protein. Here is the MLL protein complexes themselves, which then go downstream to methylate their target sites in histones and turn on oncogenic growth genes, so such as these HOX genes I was referring to a moment ago. So we think this is the pathway. Now, we can then ask, is there high MLL expression in human cancers with gain-of-function P53? So now we can turn to databases of human cancer that have been sequenced, these atlases, and we predict that there will be high MLL expression in cancers with this gain-of-function P53. And indeed, that's what we see here. It's brain glioma, a particular type of brain cancer, as I was talking about a moment ago. So we see that in gain-of-function P53 cancers, we see a high level of MLL1. And whereas in other cancers that have wild-type P53 or null P53, we see lower levels of MLL1. And indeed, if we look at all cancers in the atlas, in the human patient, in these sequencing atlases, we see that all cancers that have greater than 5% gain-of-function mutant P53, all of them have elevated MLL1 and MLL2. Okay, so now I've been telling you the idea here is that we can get to drugs, get to that these are these enzyme complexes, here's MLL1, represent nice drug targets. So others have developed drugs that interfere with these complexes. I don't want to get into the detail, but a couple of different groups have developed different drugs that target this complex and disrupt it, this MLL complex. And indeed, when we use these inhibitors to these two different locations within the complex, we see that in a dose-dependent way, these drugs lower growth, very similar to the genetic knockdown of MLL that I showed you in a couple of slides ago. So that's very interesting that, that's the case. So prevalent mutant P53 mutations co-opt epigenetic pathways to drive tumorigenesis. And the way we think of this is that P53 gain-of-function cells are addicted, if you will, to epigenetic activation. That's a phrase that's used in the cancer world, addiction to various types of mechanistic disruption. And the idea then is that tailored combinatorial epigenetic therapies may be effective against these many common cancers that are driven by gain-of-function P53. So the sorts of questions that we're interested in is, will these inhibitors against enzymes provide, growth inhibition? These MLL1, these enzyme inhibitors, these complex inhibitors, and then we see these gain-of-function P53 driving other epigenetic pathways, a particular type of promoter, acetylation, and some other types of modifications. I don't want to get into it, but these represent possible ways to get into combinatorial therapeutics if we could target multiple pathways. And you might ask, well, these are, these modifications are required for all sorts of gene activation, but if we can get into low dose, let's, for example, some sort of, low dose usage of these inhibitors in combinatorial ways, we might be able to attack these cancers without undue side effects.
You would predict that an inhibitor of an MLL1 would act better on a mutant P53 tumor than on a wild type.
That's right. By the data I just showed you, that is the case.
Have you tested it?
We have not, so it's a very important question going forward. It's loss of gain of function P53. Right. It's a phenocopy of loss. So a reduction of gain of function P53 or reduction of MLL1 both lead to lowered growth of these specific cancers with gain of function P53. Yeah. Right. That's a good question, whether disruptions of MDM2 binding could, I mean, people have been using that in, it's a good question. We have not looked, we haven't addressed whether Nutlin, which disrupts MDM2 binding, would be combinatorially effective with, you'd actually expect it to go the opposite way.
In a mutant P53 cell, it isn't making much MDM2 at all because
That's right.
it's a P53 regulated gene.
That's true.
So the question is a little less urgent than
yep.
it would be otherwise.
Mhm.
But it does make a little, and it does degrade the mutant as well as wild type. But it's really so small amount. It's not a,
Yeah.
it wouldn't be a big effect if you interrupted it.
All right, let me go on to the next little vignette. This is on brain epigenetics, and it's going to be a presage the next talk you're going to hear, a metabolic epigenetic axis in memory, behavior, and addiction. All right, so we've been talking about these enzymes that, that lay down acetylation or methylation. Well, all of these enzymes require cofactors. So in the case of histone acetyltransferases, it requires a high-energy donor of acetyl groups, acetyl-CoA, just as SAM is a high-energy donor of methyl groups to histone methyltransferases and DNA methyltransferases. So we became interested in this idea of cofactor regulation, and I'll tell you why now. So we largely think about all of these cofactors being made and in the mitochondria through all the enzymes that are located here and all the pathways that are located here to make all these acetyl-CoA and other factors. But it turns out that some of the enzymes that make these cofactors are present in the cytoplasm and even in the nucleus. So here's an example of an enzyme that we've become very interested in, acetyl-CoA synthetase 2. It uses acetate and CoA to make acetyl-CoA. Very simple reaction. Okay? So in terms of cancer, ACSS2, this is a paper from Ben Tu's lab from 2014. It's very important in cancer. It's highly expressed in human tumors. I don't want to go through this data. Tumor development is suppressed in an ACSS2 null mouse. So it's very important in cancer through it. And what led to our interest in the brain is that we found that ACSS2, looking at these Allen Institute brain atlases, is highly expressed in developing mouse brain and in the adult human central nervous system, in fact, in the hippocampus, which is one of the regions of the brain very important in learning and memory. So that led us to ask a number of questions. We know that we need very rapid changes during learning. Does the epigenome regulate learning and memory? How are neurons regulated? And how do neurons respond dynamically and quickly during learning, yet they maintain memories over a lifetime? All right, so I'll start with this simple picture of a neuron. Here's the nucleus. Here's the synapse. Beautiful work from Eric Kandel and Paul Greengard 30 and more years ago showed that signals received at the synapse are transduced through the axons into the nucleus where transcription occurs and where gene expression occurs. Well, it turns out that one important element of this gene expression is a histone acetyltransferase called CBP. CBP is a histone acetyltransferase. It's a very interesting one. It arose with eukaryotic multicellularity at the same time that you see enhancers coming up. Enhancers are gene regulatory regions in the DNA that are, there are only about 15, 20,000 genes in the mammalian human genome, but there are hundreds and hundreds of thousands of enhancers in the human genome. So enhancers are critical to regulating these small number of genes through development. And indeed, this histone acetyltransferase was then duplicated at the time of vertebrate radiation to two different enzymes, CBP and P300. And these mutations in these enzymes are seen in intellectual disability syndromes. Well, here's the CBP P300 structure, domain structure, and I just want to point out here's its hat domain. It has a lot of these reader domains to get it to the right enhancers, and it has transcriptional factor binding domains as well to also get it to the right genes. All right, so here's the, I didn't show you this, but this is the classic idea of the way these hats work, that there are transcription factors that bind to the DNA at two enhancers, such as CREB, and they recruit through protein-protein interactions these enzymes, histone acetyltransferases, and they then lay down these acetylation around the promoters, and that leads to RNA polymerase binding and transcription to ensue. So this is the classic view. And I'll just mention that CBP acetylates a number of sites, lysine 27 in histone H3, lysine 9 in histone H3, and a couple of sites in histone H4. These are key sites in enhancers and promoters to activate genes. All right, so in neurons, what we think, given what I've told you so far, what we've hypothesized is that this classic gene activation pathway, transcription factor binding, recruiting the hat, may not be dynamic and fast enough. So we thought maybe there's a direct connection at genes between the enzymes that generate acetyl-CoA as a key metabolite and chromatin modifiers. So this is work we published a couple of years ago. We also, I'm not going to be able to talk about it, but I can tell you what we have in press now. Interesting story that coming out of this. All right, so we started studying this first in vitro in cell culture, and then in vivo that I'll show you in a minute. So we started studying in a mouse, a particular cell line that differentiates from sort of pre-neurons into neurons, into neuron morphology, sending out these processes, and these cells become excitable. And so we studied the mechanism of ACSS2. The first thing that we saw, so this is this enzyme that makes acetyl-CoA, is that this enzyme from undifferentiated into differentiated cells becomes nuclear localized. So DAPI staining stains the DNA in the nucleus. You can see that in undifferentiated cells, ACSS2 is mostly in the cytoplasm. But when you differentiate these cells to DAPI again, you can see that it just hones right in on the nucleus. So this acetyl-CoA producing enzyme becomes nuclear localized upon differentiation. So our question was, could metabolite biosynthesis be localized right to chromatin, right at the genes where you need to generate histone acetylation to activate genes? This would allow a really rapid response to, say, learning. And could that happen via chromatin-bound ACSS2? So now you're looking again at more CHIP, chromatin immunoprecipitation. Here are the genes we're looking at, CAM kinase 2, very important kinase involved in signal transduction, as I was telling you earlier. Here's what, just look at this one. This is H3 lysine 9 acetylation, and you can see that in undifferentiated cells it's low, and at the promoter you can see in differentiated cells this acetylation goes up. And here's our main question: what about ACSS2, this enzyme that makes acetyl-CoA? And now you can see in undifferentiated cells very low levels, but in differentiated cells you can see that it goes up. Okay, so genome-wide, I'm just showing you a genome browser of this very crucial gene. Genome-wide, ACSS2 binding correlates with increased histone acetylation at all the genes that have to be activated during learning. And this is all in this cell culture model. All right, so now we go in vivo and we ask, what is the function of ACSS2? Could it be involved in learning? So now we need a learning task. We're going to ask this question in mice. And we're going to use a memory task called object location memory. You bring the mouse into this arena. There are 3 objects. You remove the mouse, and 24 hours later, you bring the mouse back. You move one object, and the mouse, clever little guy or gal, can spot the object that's been altered and favors that object to explore. Now we can knock down ACSS2 using a surgical method to knock it down in the hippocampus, this region that's very important in learning and memory. We knock it down in the dorsal hippocampus that is known to be involved in this type of learning. We allow the animal to recover, and then we subject it to this test. We show that ACSS2 is knocked down in the dorsal hippocampus, but not the ventral, so it stays quite localized. And now we do the experiment. So on training day, we have the ACSS2 knockdown in black versus non-specific knockdown in gray. And what you can see on training day, the mice explore the objects exactly the same, no difference. Here are the objects, here's the actual arena. So there's no general deleterious effect, and we have a lot more knowledge about this enzyme. Pardon me?
Sorry, what is the exploration index?
This is the just a quantitation of exploring each of the objects, so it explores each object exactly the same.
The higher it is, the more.
Yeah, the higher it is, the more it's hanging around each object. It's all done with imaging and quantification of the exploring. All right, so we take the animals away, we bring them back in 24 hours later, we move one object, new location is the altered object, and now we re-quantify, and now you can see in the control knockdown, the animals favor the higher exploration index, favor the new location, whereas the knockdown ACSS2 mice do not favor any particular object, all 3 the same in black. So hippocampal ACSS2 expression is required for object location memory. So I'm going to just summarize the work here. This metabolic enzyme ACSS2 is bound to chromatin. It's bound, in fact, we can see by co-immunoprecipitation assays that it's physically associated with CBP, but we don't know if it's direct. We're testing that now, the mechanism of interaction. And we find that it regulates neuronal gene expression to facilitate learning and memory. So in response to learning, it uses acetate, it makes acetyl-CoA, and then it feeds that to CBP, which then in turn acetylates the histones to turn on these immediate early genes. So that's the overall mechanism. So the interesting questions that we think come out of what we call on-site production of acetyl-CoA is how broadly does this occur in the brain, other regions as well? Is it regulating other behavioral and memory pathways? And where is the acetate coming from? Is it coming from local histone acetyltransferases turning over acetate? And we're very interested in this mechanism, whether there are different types of histone acetylation, some that provide a storage of acetylation, others that are involved specifically in activating genes, or could it come from exogenous sources? Yes.
So was there any specific reason that you looked first to these locations?
These particular genes?
Why you looked to the memory, this sort of spatial memory?
Why did we use that task?
Yeah, was there any specific reason
Yeah.
for that?
We just used that task. It was a simple task that our core, behavioral core was, we could do. Since then, we've done other tasks requiring this part of the hippocampus, and we see that it's required. We're very interested, I'll get to this at the end, other types of memory that's more health-related, such as unwanted memory and addiction and fear memory. So we see that it's also involved in that sort of memory. So it's not specific to this type of memory. Yeah?
So is it known if in humans the regulation of this kind of genes or this epigenetic tolerance give rise to also problems in memory, or those that we know exist, this kind of diseases?
Do we see mutations in this gene in disease? There isn't a lot of sequencing in the types of diseases we're interested in. For example, post-traumatic stress disorders, PTSD. What little we've been able to see, such as in addiction, alcohol, we do see some changes in the levels of this enzyme, but we don't know a lot about it yet. We're very interested in that.
Kids with problem on learning, for example, kids that have like deficit on the attention.
In intellectual disability syndromes, yeah, we have not seen that in this case. Okay, so again, in human therapy, we're interested in ACSS2 inhibitors. We think they may be ameliorative to diseases of unwanted memory, PTSD and addiction, and also may be ameliorative to cancer. All right, so now I'm going to give you the last little section on aging and neurodegeneration. Here we think that histone acetylation may protect the cognitively normal aging brain. So this is a different sort of idea. So we're very interested in aging. This is the aging course of Diego Rivera. These are self-portraits over his lifetime. He's brilliant Mexican muralist. So the question we're interested in are deterioration and disease inevitable during aging. So I'll just say as a way of introduction that that the news is not all bad. If you look at the historical change of human lifespan, the life expectancy at birth between years 1900 and 2000, the maximum, the life expectancy, I'm looking at the time and thinking I gotta go faster. The life expectancy, the maximum lifespan at birth is about the same at birth. So it's set by genetic factors, whereas life expectancy at birth has actually doubled between 1900 and 2000. So that says, in very broad terms, that the environment must impact lifespan and age-associated disease. Part of it is infectious diseases, but that could also be epigenetically mediated. It probably is a lot of it. It's also antibiotics. I mean, there are a lot of elements that come in. That's why I say, broadly speaking, in the environment. All right, so why do we think some of it's epigenetic? So the lifespan can also be remarkably variable, even for highly related animals. So the mouse maximum lifespan is around 2 years, and the very related naked mole rat is about 30 years. So, well, it's a lot uglier, but it does live more than 10 times longer, so that's interesting. Now, here's my little ant element here. So as Arnie mentioned, we're interested in ants. This is one of the species we study. In this case, the ant queen and the ant workers are all female. They have identical genomes. The ant worker lives about a year, and the ant queen can live in the species about 30 years. So again, with exactly the same genome, these are different, exactly the same genome, you can get a more than 30-fold difference epigenetics, okay? Wouldn't we like that? So epigenetic plasticity of lifespan provides hope for improvement of age-associated disease. And improving a lifespan might help a constellation. This is sort of the, you know, the mantra in the aging field now. It might affect a constellation of age-linked disease. So the idea, if you can get at some of the central mechanisms involved in aging, you might be able to affect a lot of diseases rather than attacking one disease at a time. Okay, so here's the little story I want to tell you about the human brain, comparing normal aging to Alzheimer's disease. This was published last year. So here, this is a very simple set of experiments. We simply did epigenomic profiling in the human brain. We used normal young versus normal older human brains versus Alzheimer's disease. Now, here, to do this kind of study, you have to, you get obviously post-mortem human brains from biobanks. They have to be very carefully annotated so you know what you're dealing with. And we cannot use the hippocampus, which I'm sure many of you know is the first part of the brain that's affected in Alzheimer's disease. Rather, we use the middle temporal gyrus because it's intact. We made very sure by various means that we had similar numbers of neurons in the study group of the normal older versus Alzheimer's disease. And I also want to mention that most work being done on Alzheimer's now compares normal older and Alzheimer's, so you don't get the change of between aging of the brain. So that's a very important element, as you'll see in a second. All right, so we decided to look at this modification. It's yet another one, the alphabet soup of modifications. This is histone H4 lysine 16 acetylation down here. It's put on by a couple of different enzymes, MOF and TIP60. We were interested in this modification because we know it's abundant across the genome. It's the first one that we did because we wanted something that was abundant. We were afraid that in Alzheimer's we would see very low levels of some of the modifications, and we wanted something that was high. All right, so we did ChIP-seq. I don't have to explain it again. Across the entire genome, we found that this modification actually increases during normal aging and decreases in Alzheimer's disease. So here's one particular gene, again, gene browser, and you can see at its promoter, you can see the signal of lysine 16 acetylation going up during normal aging and being reduced in Alzheimer's disease. All right, if we look genome-wide now and we collect all of the peaks and we just sum them in normal, cognitively normal old, we see that indeed in cognitively normal old, this modification goes up and it decreases in Alzheimer's disease. And if we now do a two-way comparison across the genome, looking at the peaks in Alzheimer's disease during aging, we find that where the peaks go up during aging, they decrease in Alzheimer's disease. And that's where most of them are. Now, if we do a three-way comparison, you guys are physicists, so this is a piece of cake for you guys. Now we have three classes of mark changes. So this schematically shows you what I mean. We have age-regulated changes. These are changes that are, these are peaks that are high in young and decrease in old and stay low, or low in young and go up in old and Alzheimer's. We have, well, disease-specific is very easy to imagine, high in young or low in young, and then remain the same in cognitively normal old, and then change in disease. So those could be driving disease. And then we have age dysregulated. So these are the interesting ones in the class that I've been showing you. So this is the particular class I've been showing you, low in young, high in cognitively normal old, and low in Alzheimer's. And indeed, if we just look at the number of peaks in each of these classes, we see that by far the most peaks that are of alteration in these three-way comparisons is this class of higher in old and lower in Alzheimer's. All right, so there's a higher level of K16 acetylation in cognitively normal brain. This suggests that this is what really interests us, that it suggests possible epigenetic protection. So how can we test this hypothesis? Well, obviously there's no testing in humans, but we can do kind of a test where we can ask whether this lysine acetylation correlates with human disease. So now, do you mind if I finish? Because I'm getting close to the end here. So now we can look at human population studies and to discover disease-linked mutations. So GWAS studies, genome-wide association studies, are looking at changes across the entire genome. And now we're looking at single nucleotide polymorphism, SNPs, across the genome. Where do they occur? Now relative to the changes in lysine 16 acetylation. So now we can look across all the chromosomes, group all the SNPs across the genome, and now compare them to where we see the lysine 16 acetylation peak shown here. So here are here's an example of a GWAS SNP in a genome browser. Here are the SNPs. Here are the acetylation peaks. Here are the genes. So now we can compare the location of the SNPs with the location of lysine 16 acetylation. And what we see is that where we see a higher level of acetylation, we see a prevalence of these SNPs. But very importantly, and this is the point I want to dwell on, where we see a low level of these SNPs associated with disease, we see the age-dysregulated lysine acetylation. So I know that's a little bit confusing, but I'm just going to summarize it, and then we can talk about it at the end. So the age-regulated and the disease-specific peaks and their correlation with the SNPs suggests that these predispose to AD. Whereas this particular class, increasing in aging and decreasing in AD, we see a lower level of SNPs associated with disease. So we believe that these are protective to AD. They're higher in cognitively normal aging. Indeed, I'm not going to tell you about it. We have a new story in review where we look at disease-specific driving, and there we see a different class of acetylation. I was saying earlier that we think there are two different classes of acetylation that have very different roles in the genome. The type that may store acetylation and the type that may activate genes. And in this case, we think the storage type is protective and the activating type is driving disease. I mean, I can get into that in detail if you like in the questions. All right, so the epigenetic model of Alzheimer's disease, the normal aging human brain displays features of epigenetic protection against mutations that occur in late onset Alzheimer's disease. This provides hope for an epigenetic drug therapeutic as a future goal. So the questions coming out of this area: do other age-associated epigenetic changes protect from AD? Which age-associated epigenetic changes predispose? I was just telling you about that a moment ago. What are the hats and can they be pharmacologically targeted? This class, the type that predisposes, are kind of a good class because those, again, inhibitors would lower them in predisposing to Alzheimer's disease. And what about other neurodegenerative diseases? We don't know anything about those yet. All right, to sum all of this up, human health and therapeutics, epigenetic regulation involves enzymes and binding proteins. These are frequently mutated or overexpressed in disease. Superb targets for drug development. I just tried to list a few for you. In cancer, in the clinic, we have DNA methyltransferase inhibitors, HDAC inhibitors, and IDH inhibitors. We have some in third stage, in phase 2, phase 3 clinical trials, BRD4 inhibitors, EZH2, MLL, and DOT inhibitors. We have some that we would love to develop, MOS inhibitors and ACSS2 inhibitors against cancer. Against unwanted memory, HDAC inhibitors are actually being used. We think that's the wrong way around. That would promote acetylation. We think we want ACSS2 inhibitors that would lower acetylation. So we're very interested in that. improved memory and neurodegeneration. HDAC inhibitors, I believe, are in clinical trials. We would like to develop ACSS2 agonists to improve acetylation, at least in one class. But I did give you a lot of complexity there because some acetylation would be negative. All right, my neuroepigenetics team, this group, I'm not going to go through this in detail. Our funding over here because there's not time. And then cancer epigenetics team and our funding here. And my lab. And I'm happy I'll just stop here and take any questions. Sorry, I went a little bit over.